Hi everyone and welcome. Depending on where you are, good morning, good afternoon. I'm Bori Darpo. I'm a cardiologist and Chief Scientific Officer of Cardiac Safety with ERT. It's my great pleasure today to present on the proposed revision of the ICH E14 guidance and also share some thoughts on the content and how I believe it may be implemented by different regulators. Following me, Dr. Kleiman will present on the proposed S7B Q&A document, which is new. So the background is the following. Based on the 2005 S7B and E14 guidances, the role of non-clinical assays have so far been mostly to make sure that NCEs can be safely taken into humans, so to ensure safety for subjects enrolled into first-in-human trials. The proposed Q&A revision of S7B and E14 suggests an expanded role. So if non-clinical assays are performed according to best practices as defined in the S7B Q&A document, they can also be used for decision-making later during the clinical development of a drug. This is more specifically addressed in the E14 question 6.1 that allows sponsors to categorize drugs as low risk if the mean effect is below 10 milliseconds, even when the study was performed without placebo, because it cannot be safely given or justified, and when a therapeutic dose is evaluated, if supported by negative non-clinical assays. And this is, as you can understand, mostly oncology. Question 5.1 obviates the need for a therapeutic study in case a twofold exposure margin versus the high clinical scenario cannot be obtained, again, if supported by negative non-clinical assays. So to give some context to this and some background, I would like to walk us back to the 2015 revision of the E14. By that time, extensive experience had been gained by regulators and by sponsors by applying concentration QTc analysis on data from therapeutic studies over 20 years, as a matter of fact. The revision was also based on the prospective IQ-CSRC study, which was performed in collaboration between industry, FDA, and the Cardiac Safety Research Consortium, and that demonstrated that small studies can detect mildly QT prolonging drugs if concentration QTc analysis is applied. And it's fair to say that today the majority of therapeutic studies are performed with concentration QTc analysis, which has substantially reduced the required sample size down to 24 to 28 subjects as compared to the traditional by time point analysis, the intersection unit test. More importantly, in many first-in-human studies, serial ECG monitoring is applied paired with PK sampling, either in SAD or in the multiple ascending dose part of the study. And as we advocate, by collecting and storing the ECG waveforms, the sponsor can make an informed decision on when and what to analyze. So this is how the 2015 revision looks like. And we will come back to both these points, perhaps mostly the first point. If concentration QTc analysis is applied to a study in healthy subjects and sufficiently high multiples of clinically relevant exposure has been achieved, then a separate positive control would not be necessary. This is really important because in the vast majority, if not all, first-in-human trials, there is no positive control. But if we then achieve sufficiently high concentrations, we can use this data without having to perform a therapeutic study with a positive control. And then secondly, the same criteria as we used in the IQ-CSRC study was brought back into the E14. So if you are able to demonstrate that your drug does not cause an effect exceeding 10 milliseconds at clinically relevant plasma concentrations, that is the same as to say that you have a negative QT assessment and carries the same weight as having a negative thorough QT study. And this is then one example of a waiver for a therapeutic study that was granted in all three major jurisdictions. This is LEMBOREXANT, a drug intended to treat insomnia. And we're looking at data from two multiple ascending dose studies. First, the standard first-in-human MAD study, and then a bridging MAD study intended to demonstrate similarity of PK between the Japanese population and the Caucasian. The left graph shows you a scatter plot with all the data pairs, and as often is the case, you don't have that much of concentrations in the high end. The right graph shows you a goodness of fit plot, where the black line is the predicted effect on QTc as a function of plasma concentration. So the y-axis is the placebo-corrected change from baseline QTc, and the x-axis is the concentrations. And we can see that the model predicts that this drug will not cause QTc prolongation. And by looking at the gray shaded area, the 90% confidence interval, we can also see that the predicted effect will be nowhere close to the 10 millisecond threshold. Now, importantly, clinically relevant plasma concentration, so LEMBOREXANT, is around 40 to 60 nanograms per mil. So we are 4 to 5-fold above what you will see in patients, including patients with impaired clearance. These data were shown to FDA and then somewhat later to PMDA in Japan and in Europe. And all three regulatory bodies granted permission to proceed into pivotal trials without performing a therapeutic study. LEMBOREXANT was approved in December 2019 in the United States with a label shown here that describes the results, but also concludes that this drug will not cause QTc prolongation. So this slide shows you the current paradigm that FDA has applied when they're looking at concentrations achieved in first-in-human study and whether a positive control will be needed. This approach, or this paradigm, is now brought into the ICH E14 document. So this is an example. If we take a drug with a 10 milligram dose being the therapeutic dose, and that dose gives you a mean Cmax at steady state of around 100 nanograms per mil. Then we look at the patient population with impaired clearance, let's say based on the drug interaction. Let's say that the drug is a 3A4 substrate, and then we're looking at the mean Cmax at steady state on the highest dose with a potent 3A4 inhibitor. Let's then say that we have an increase of the Cmax up to 300 nanograms per mil. Then what is here called the supratherapeutic dose would be higher than the 300 nanograms per mil level, whereas really high concentrations, or the concentrations needed to obviate the need for a positive control, would be twice the 300, so 600 nanograms per mil. This is generally the approach that has been used by FDA, but it's not all. It has served as a guidance for other regulators, but it's not yet written into the guidance, and its interpretation has varied from one regulator to another. This is a slide also shown at the public webinar showing the distribution of studies intended to demonstrate that a drug does not have much of a QTc effect. So around 40% of the studies between 2016 and 2020 were therapeutic studies, meaning studies with a positive control and a supratherapeutic dose or / concentration. Around a third of the studies were 5.1 studies, so first-in-human studies on which concentration serial ECG monitoring was paired with PK sampling. And 6.1 studies are the same as oncology studies, roughly a quarter of the studies. Now, this is an average over the years 2016 to 2020, whereas this is an ongoing process. The proportion of therapy studies are probably even lower today as compared to this average over 5 years. So 5.1 refers to studies in healthy subjects where you cannot achieve sufficiently high concentrations. And 6.1 refers mostly to oncology studies. And this is the scope of the current E14 revision. 5.1 allows the sponsor to pair their clinical data with what is termed a double negative non-clinical assay to support a lower margin between the achieved exposures in the first-in-human study and the clinical exposures in order to obviate the need for positive control. Whereas 6.1 supplements clinical data with a double negative non-clinical assessment to support alternative clinical study results from oncology studies to claim that the drug has a low likelihood of proarrhythmic risks. So let's start with 5.1. And it's important, I believe, to point out that the text says when sufficiently high multiples cannot be obtained for reasons of safety, tolerability, saturating absorption, then a non-clinical integrated risk assessment can supplement the clinical data, meaning that if you have a negative hERG assay and a negative in vivo study, and you have and you cannot achieve high concentrations in your first-in-human study, the non-clinical data would help you arguing that you will not need a positive control. Up until today, it has often been the practice, at least from FDA's perspective, to ask for a therapeutic study when sufficiently high concentrations have not been achieved or cannot be achieved in the first-in-human study. So some thoughts and consideration from our perspective. The current practice, as I said, has been to request a therapeutic study with positive control in cases where high concentrations has not been achieved or cannot be achieved in the first-in-human study. The proposed change will therefore to some extent lower the requirement. That is, it will reduce the number of therapeutic studies and enable acceptance of robust, high-quality ECGs using concentration QTc analysis to be supplemented by non-clinical assays to demonstrate that a drug does not cause clinically relevant QTc prolongation, even in the absence of a positive control. And again, I think it's important to point out that the text says that when sufficiently high concentrations cannot be obtained. On the other hand, we know that high concentrations is really important and actually drive the power of concentration QTc analysis to demonstrate that a drug does not have much of an effect. And I would like to share one example of this. This is a study we did some years ago in collaboration with J&J. And the blue line with blue solid and hashed lines is the concentration QTc relationship when the highest dose, the 150 milligram dose, has not been included. And we're looking at the QTc interval on the y-axis and concentrations on the x-axis. And we can see that when we're looking at doses up to 90 milligram in a multiple ascending dose study, the model predicts that this drug will not cause QTc prolongation. Then a specific cohort was added with a higher dose, the 150 milligram multiple ascending dose was added. And then we can see that the QTc effect is teased out. So the red line with the hashed confidence intervals are now the predicted effect when also the 150 milligram dose concentration data are added. So this example clearly demonstrates the value of adding high doses / concentrations. And we also know, based on experience from the IQCSRC study, that adding high concentrations really drives the power of the analysis. So it is important to achieve these high concentrations. And whenever that is feasible, that seems the most appropriate approach. I would also like to note that the requirements for non-clinical assays are not very clearly defined. I think it's somewhat surprising that even though we have more than 20 years experience of hERG, GLP hERG assays and therapeutic studies, sponsors have not been able to agree on clearly defined criteria for claiming that a hERG assay is negative. Many sponsors still use the 30-fold margin, which is described in the Redfern paper, but this is now not taken into the E14 guidance, and Dr. Clymer will further elaborate on this. It should also be noted, in my view, that to claim that that an in vivo study is negative, you do not need to have supratherapeutic concentrations, so not concentrations higher than clinically relevant concentrations. I believe that lack of clear and agreed upon criteria is likely to result in different interpretations across regions. That is, we will not achieve harmonization by avoiding very clear and agreed upon criteria. And I think, and we think, that it's much based on clear criteria in the clinical area that we have been able to achieve an harmonized approach across regulators, at least to a large extent. That is, the sponsors will know what they can expect. So this slide from Dr. Garnett's presentation on the public webinar describes the 5.1 scenario as two pathways to waive the positive control in a clinical study. I think the key point, which is not acknowledged here in this slide, is that 5.1, according to the text, applies to drugs for which supratherapeutic concentrations cannot obtain. It's not a general approach to say that the positive control must not be required. Also, the chart shown here does not provide an insight as to whether high concentrations were not achieved. We often see that the doses were not really pushed in the SAD or MAD study. But as I said now repeatedly, 5.1 refers to the scenario where you cannot achieve sufficiently high concentrations based on limitations in terms of safety, tolerability, or saturated absorption. It is therefore, in my view, not clear to which extent the proposed 5.1 will further reduce the requirement, the number of surrogate studies. But we can certainly expect a continued debate between sponsors and regulators in regard to this specific item. So let's then cover 6.1, which mainly addresses oncology studies. And this is just an extract from the text. Much of this already exists in the previous revision. And for oncology studies where we typically cannot justify or cannot give placebo, and where often the therapeutic dose is the study dose, we don't have a supratherapeutic dose, then we are today asked to take as many components as we can into a patient study. Now, the revised E14 document proposes that if these data are supplemented by negative hERG assay and a negative in vivo assessment, and Dr. Kleiman will elaborate on the somewhat different requirements in this scenario as compared to the 5.1 scenario. But if we can supplement our clinical data, and if we see that we don't have a QTc effect in the patient trial, then we can claim, that the sponsor can claim, that the drug has a low likelihood of proarrhythmic effects. Another example here that is given in the guidance is drugs that has a substantial effect on the heart rate, so either increasing or decreasing the heart rate by more than 20 beats per minute. So my thoughts on this is that this is mostly about oncology. These studies are often conducted in patients, often without placebo and with a therapeutic dose. So this is mostly oncology. We also see some of this in other areas where a therapeutic dose in patients really cannot be given to healthy subjects. Dopamine agonists and some neuroleptics, as an example. I believe that example with a pronounced heart rate effect is poorly chosen, should be better to taken off from the guidance. In the view of many regulators, this type of drug that causes a substantial heart rate effect to this extent will have to undergo phase 3 ECG monitoring anyhow. And please bear in mind that the E14 document is about demonstrating which drugs should undergo ECG monitoring in phase 3. It's really the role of the therapeutic study to determine this and not the labeling as such. I think it seems correct to claim that a drug that increases the heart rate by 20 beats per minute will not cause proarrhythmic events based on delayed repolarization. On the other hand, it may well trigger coronary ischemia or worsen heart failure, and thereby cause life-threatening arrhythmias on this basis. Also, a drug that causes a reduction of the heart rate that is having a strong negative chronotropic effect is likely to cause clinically significant bradycardia. It may trigger sinus pauses and potentially AV blocks. I therefore think this example is torsades-centric and ignores other mechanisms by which a drug may be pro-arrhythmic. The current FDA approach to studies in oncology is now brought into E14. So this is standard practice from FDA's perspective, and this is now brought into E14. So if you can demonstrate that your drug does not have an effect, defined as the upper bound being below 90%, the upper bound being below 10 milliseconds, then the drug is unlikely to have an actual mean effect as large as 20 milliseconds. So if we run a study with an oncology compound without placebo and with a therapeutic dose, and we can demonstrate that we can exclude an effect above 10 milliseconds, which is the upper bound is below 10, then this is what will go into the label. I think it should be noted that depending on the mean effect, this may or may not come with ECG monitoring in clinical trials, and also some statements in regard to whether patients on other drugs that causes QTc prolongation can be incorporated, and whether the drug can be given to patients at risk. The novelty here is that 6.1 questions 6.1 describes a process by which a sponsor can claim that a drug has a low likelihood of proarrhythmic effects. And for this, all these three criteria should be fulfilled. First of all, we should have a negative hERG assay and a negative in vivo assay as performed following best practices. Secondly, the QTc effect should be below 10 milliseconds. Please note that it's written this way, generally defined as change from baseline QTc greater than 10 milliseconds. If that is not seen, and we have confidence in the strength of the clinical data based on the confidence intervals, then this comes with a claim that then the sponsor can make the claim that this drug is unlikely to have pro-arrhythmic effects. Then, of course, a cardiovascular safety database that doesn't suggest anything alarming would also be required. To summarize, negative non-clinical assays, demonstration that the mean effect is lower than 10 milliseconds, the mean effect, and also then a database that does not suggest that we see anything alarming in terms of cardiovascular adverse events. So if 1 and 3 are fulfilled, then it can be claimed that the drug has a low likelihood of proarrhythmic effects. And this is specifically where the E14 S7B implementation work group is asking for public comments. The main reason for this is that there is a disagreement in the group because this is a novel criteria that is brought into E14. It should be noted, and Dr. Kleiman will elaborate on this further, that the definition of a negative in vivo study is not exactly the same in 5.1 and in 6.1. The main difference being that in 6.1, the in vivo study should be powered to detect the QTc prolongation similar to the effect in the clinical therapeutic study. It should be noted that the novel criteria is looking at the mean QTc effect that should not be more than 10 milliseconds. It comes with a certain qualifier. It says that the strength of the clinical ECG data depends on the upper bound of the two-sided 90% confidence interval. This is a new criteria. It clearly, as I said, the most debated topic among regulators in this group. This would then enable the sponsor to claim that the drug has a low likelihood of proarrhythmic effects. And that this would then translate, but it's unclear how this would translate into decision-making in terms of pivotal trials. I think it must be emphasized that ICH does not regulate or guide labeling. This is outside the scope of ICH. So the main questions for regulators and for sponsors is, how should we see low likelihood of pro-arrhythmic effects? Does that mean that the drug can be taken into pivotal trials without ECG monitoring, without precautionary statements in regard to concomitant medications with other drugs that has a QTc effect, and without cautioning patients with hypokalemia or risk factors for torsade to be included? These are important details that are lacking, and we're left without a clear answer on this topic. So the example given during the public seminar is a drug that causes, first of all, is of course double negative when it comes to non-clinical assays, and then has a mean effect of 4 milliseconds with an upper bound of 12 milliseconds. And the question is then, can the integrated QT assessment support low risk for QTc prolongation under Q&A 6.1? And FDA's answer is yes here, since non-clinical assays were conducted according to best practices. But let's take a more challenging example, still of course within the definition of the proposed E14 wording. Let's say that a drug has a change from baseline, mean change from baseline of 8 milliseconds. And let's use the same width of the confidence interval. So the upper bound would be 60 milliseconds. In my view, it seems very likely that regulators will see different on this, and there will be clearly differences in opinion in regard to whether this drug can be taken safely into pivotal trials without ECG monitoring. And also, it seems likely that there will be differences in opinion across regulators, whether the drug can be given safely with other drugs known to cause QTc prolongation and to patients at risk for pro-arrhythmic events. And thereby, the desired level of harmonization is not achieved, and this is much based on a new criterion that regulators seemingly do not agree on. So if I am to summarize the proposed revision of E14, so FDA describes, at the public seminar, FDA described the E14 revision as a value proposition. The cost being that sponsors in specified cases can perform best practices, HERG and VIVO studies in a standardized way, as described in the new S7B Q&A document. And the value would be that this would further reduce the number of therapeutic studies. If supported by negative non-clinical assays, then E14 question 5.1 will obviate the need for a therapeutic study in case a twofold exposure margin versus high clinical scenario cannot be obtained. And 6.1 will enable the categorization of drugs as low risk, as an example in oncology studies, if the mean effect is below 10 milliseconds. And in my view, the new criterion in 6.1, the mean effect below 10 milliseconds, and the lack of details in the description of a negative HERG and in vivo studies will lead to differences in interpretations across regulators and thereby counteract the desired harmonization. Thank you so much. And before taking questions, I would like to hand over to Dr. Kleiman. Dr. Kleiman, please.
Thanks, Dr. Darpo. And I'm Bob Kleiman. I'm a cardiac electrophysiologist and Chief Medical Officer for ERT. And today I'm going to tackle the S7B draft question and answers. And I'll note this is the very first Q&A since the release of the document in 2005. So for 15 years, there really hasn't been a lot of elucidation of changing technologies, thoughts about best practices. And the Q&A has 4 questions, which at a high level address the topics first of integrated assessment of risk, second, best practices for in vitro studies, best practices for in vivo QT studies, and finally, principles of proarrhythmia models. I will not go into all the details of each of these, or we would be here for several hours, so I'll try to highlight this. But first, I want to say the reason this is as important is it's going to change the balance between non-clinical and clinical cardiac safety data for use in regulatory decision-making. Since the release of S7B, non-clinical cardiac safety studies have usually been used primarily for go-no-go decisions or to inform the design of first-in-human studies. And they've been viewed as good to have, the negative study being good but not sufficient to alter the ultimate clinical decision-making based on the clinical data. And I think that may be part of the reason that best practices for the non-clinical cardiac studies haven't really been clearly formulated. And as a result, they're not performed uniformly across all sponsors and all labs. What the proposed revisions to both E14 and S7B will do is make the non-clinical cardiac data more of an equal partner to the clinical data when making an integrated assessment of proarrhythmia risk. I'll point out this is going to raise the bar for conducting, design, and even reporting of the data from these non-clinical cardiac trials. So question 1 deals with the integrated assessment of risk. And at a high level, it discusses using the non-clinical data to inform the design of first-in-human studies, as well, and this is the new part, to complement the E14 data for an integrated assessment of torsade risk. So in the past, one might have done these non-clinical studies truly as preclinical studies before starting human trials. Now, they may truly not be preclinical because you may want to repeat earlier non-clinical studies at a later time based on the need to use this data for decision-making, as in E14 questions 5.1 and 6.1, and then we're having the requirement that best practices be met. This answer to question 1 discusses a bit about hERG safety margins, about the need to assess both apparent and metabolites, and there's some discussion of the exposure required for the in vivo QT studies. It is not as precise as I would like. It mentions that the exposure tested should be at least one that covers the high clinically relevant exposure. But given the importance of high concentrations in small studies, as Dr. Darpour just described, I think it can be argued that it is probably much better if you can obtain a truly high supertherapeutic exposure in your non-clinical studies. And I'll also note that I suspect that different regulators will have different requirements for the high exposures that they would like to see in our non-clinical trials. Question 1 addresses the double negative non-clinical package that is mentioned in the E14 update. And that means first a negative hERG study, which is loosely defined as a hERG safety margin that's greater than the safety margin for a series of drugs known to cause torsade. It doesn't really define what the adequate safety margin is, whether that's 30, as suggested by the Redford paper, or should be left up to each sponsor is not clear. It's also not clear which torsadogenic drugs should the new drug be compared to. The second criteria is a negative in vivo QT study, with all the caveats about adequate sensitivity, high enough exposures of the parent compound, as well as of human-specific major metabolites. And there's a comment, and this will repeat several times, additional non-clinical studies, such as Purkinje fiber studies, cardiomyocyte studies, are probably only needed when you are using the non-clinical data to support E14 5.1 or 6.1 pathway, and if there are factors that make the patch clamp study and the in vivo QT study either difficult or impossible to interpret, such as metabolites, heart rate changes, etc. The Q&A mentions some other factors that may modify torsade risk. Some of these are non-hERG mechanisms for QT prolongation and risk of torsade. Others are factors that may mitigate. QT prolongation related to hERG channel block. So you may have block of other repolarizing currents, or you may have block of inward currents, such as late sodium current or L-type calcium current. These may mitigate the QT prolonging effects of hERG block by the compound, but on the other hand, they may pose their own risks, QRS prolongation or LV dysfunction. We also know that some drugs prolong QT and produce torsade, not by blocking the hERG channel directly, but by affecting the trafficking of hERG channels. We know that metabolites may be responsible for hERG or other ion channel block. And we also know that there are non-ion channel related mechanisms for producing QT prolongation. So these are important things that I'm glad to see the ICH reminding us of. And here is an example of a mixed ion channel blocker. This is DOLASETRON. And in the top panel, which shows concentration versus change in baseline for QTCF, placebo corrected, there's a very clear relationship between QTC and plasma concentration. And that would suggest, oh, this is a hERG blocker that is potentially torsadogenic. But in the lower panel, we're looking instead at J to T peak. And here you see that as concentration increases, J to T peak does not increase. So this is a mixed ion channel blocker, no JT peak increase. And in the CSRC consortium study, PR increased by 16 milliseconds and QRS increased by 5 milliseconds because DOLASETRON also blocks the late sodium and L-type calcium currents. Question 2 deals with best practices for in vitro studies, meaning primarily patch clamp studies or cardiomyocyte studies. And it starts off with the reminder, the best practice considerations are only intended to apply when you're using the non-clinical data to support an integrated assessment of proarrhythmic risk as described in E14, 5.1 and 6.1. These are not intended to be guidelines that must be followed for all non-clinical studies conducted for new compounds. You may use the standards that you're currently using, or you may decide to always use best practices for your patch clamp studies. That's really up to you, and it's not obligatory. In terms of best practices for ion channel studies, there's some discussion of primary endpoints, the use of physiologic versus room temperature, how to assess ion channel recording quality, the verification of concentration in your bath, the use of positive and negative controls, and even some suggestions about how to present data in reports. And then there are best practice considerations for myocyte studies. They're generally at a relatively high level, although they do specify that one should be quite clear about the source of the cardiomyocytes, the protocol used, validation of viability, and details about the assays for cardiac risk that you're going to be performing. Question 3 discusses best practice considerations for the in vivo QT studies. And overall, these are really high-level discussions and don't have a lot of specific details about particular best practices, no discussion of ECG collection, which leads should be used, how ECG measurements should be performed, and most of all, what is the threshold of concern? What is a negative in vivo QT study? And I won't go through all the details. There is some talk about the exposures required, but as Dr. Darpo mentioned, while it isn't even clearly stated that one must have a super therapeutic exposure, it seems reasonable that one might want to have, if possible, an exposure that's multiples of the high clinically relevant exposure. So a little bit of discussion about QT correction for heart rate, which is quite welcome. little bit of discussion about the use of reports to present the data that regulators would like to see. And unfortunately, if you'll remember in the E14 6.1 section, there was talk about a study having the power required for a clinical thorough-QT study, but it's really not elaborated about what that means. There's some discussions finally about proarrhythmia models and how they might be used as part of an integrated assessment of risk. But again, relatively high level. So to summarize, part 1 presents some useful high-level concepts about using non-clinical data to supplement the clinical ECG safety data when trying to assess proarrhythmia risk. That's E14 5.1 and 6.1, and 7B discusses some of the details of how to conduct such studies. Part 2 is about best practices for in vitro studies, and there's some important concepts, but I would say limited details about cardiomyocyte studies. A little more about the patch clamp studies, but still, there are some details that are not made clear. I find that part 3, the discussion of best practices for in vivo QT studies, presents some high-level concepts, but very limited details about design, performance, assay sensitivity, required exposures that I think really are necessary for us to understand. And then finally, principles of arrhythmia, proarrhythmia models are discussed, again, as high concepts, but without a tremendous amount of detail, without suggesting which models might be the most appropriate to use. I'll reiterate the point I made earlier. When should S7B best practices be used? Now, you're always welcome to use best practices all the time, but that isn't absolutely necessary. These are really only required if you're going to use the non-clinical data to supplement the clinical data as part of an integrated assessment as described in E14, 5.1, and 6.1. And that may mean that you continue to do non-clinical studies as you are currently doing them, and only if you find that as you're late in phase 2, you realize that you are going to have to follow E14, 5.1, or 6.1 pathways. At that point, you might want to go back and repeat your patch clamp studies or your in vivo QT studies according to the best practices. So plan on doing it in a graded fashion. So some of my thoughts and some considerations about this. First, this is the first set of question and answers for S7B, whereas E14 is now on the 5th set. That's great, but while it describes some high-level concepts, we do need more details. And in contrast, in the clinical cardiac safety realm, we have resolved a tremendous number of details about how to perform thorough QT studies and first-in-human QT studies through the series of E14 question and answers, as well as 15 years of regulatory consultation and interactions. I think the more detail about the best practices will be useful for two purposes. First, to better standardize how these trials are conducted throughout the industry. And second, and this is one that's close to me, we need to understand these best practices so that sponsors can go and look at their non-clinical data in detail and really understand if it's going to be of acceptable quality to be used to support the E14, 5.1, and 6.1 strategies. Right now, we don't always see all of the details or understand all of the details of how the patch clamp studies are done or how the animal ECG data were collected. But I think that's going to be much more important going forward. So my final thoughts on S7B are, this is a very important first step, and we should all congratulate the working group for doing this. And finally, remember, E14 and S7B specifically are addressing the risk of drug-induced torsade. and indirectly other ion channel effects. But torsade is only one of many different cardiac side effects that new drugs need to be evaluated for. So thanks. That's going to close out our presentations on the E14 and S7B question and answers. And now I'll close with a few thoughts in summary about the combination of the two question and answers. I will start just with a very brief summary of what I think that we should take away from these Q&As. First, I think that this is a huge advance. For the first time since 2005, we have both clinical and non-clinical discussions that are actually paired together. And it's a stepwise progression. E14 updates describe how you would do trials, how you would use the non-clinical data when you can't achieve the high clinical exposures that one would like, or when typical TheraQT studies are not feasible. And then, switching over to S7B, this describes at least the framework for how the high-quality studies needed to support the clinical data would be obtained. I look forward to, over the next few, I hope it's months, but perhaps years, having more details about the specifics of the non-clinical studies, so that those of us who are asked to review non-clinical data, even though we're not in the non-clinical realm, will have some ideas of the standards we should be looking for. So with that, I'm going to open this up for questions. And here's a question for you, Dr. Darpo. How far is the FDA accepting of other approaches than classic TheraQT studies or concentration QT studies, such as approaches like bias analysis when ECGs collected in trials with a robust clinical conduct and high sample size?
Thank you, Dr. Kleiman. I haven't seen any formal or public comments on this, but I know that the bias sensitivity method at least was implemented in the ECG warehouse some years ago, and this was described at a DIA meeting we had hosted by CSRC and FDA. So there is some value in this. I also know that some regulators look positively on using the food effect as providing assay sensitivity. So I think this is more on the level of providing additional reassurance and confidence in the data. It's not exactly replacing a pharmacological positive control, but it provides reassurance. Both the method bias sensitivity test and the food effect on the QTC interval can be used to increase the confidence in the data in case you don't have moxifloxacin in your study.
Thanks. Here's a question about the non-clinical side of things. Are non-clinical double negative results required for macromolecules, monoclonal antibodies, very large peptides? And the answer is discussed in some of the earlier E14 Q&As. Monoclonal antibodies and other very large macromolecules aren't really expected to prolong QT directly by interacting with the hERG channel. They might do so through other mechanisms, but the non-clinical patch clamp studies are almost never done for monoclonals. And the animal ECG studies, I'm not sure that they really would be considered nearly as relevant as they are for small molecules. Gloria, here's a question for you that I think is on everyone's mind. The E14 updates describe the 5.1 pathway when adequately high multiples of the high clinical exposure can't be achieved. But how about if you've done your SAD and MAD studies and you didn't get to those double the highest clinical relevant threshold, but you didn't reach the MTD yet? What should a sponsor do in that case?
I think the question is a good illustration of where we will see a continued debate between sponsors and regulators, but also importantly, in my mind, or in my view, that different regulators will interpret this differently. It may well be that some regulators would accept data and would accept that non-clinical data can supplement the clinical data in that scenario, and therefore not require a study with a positive control. Whereas other regulators will just conclude that the sponsor haven't really tried. So I think that's a good illustration of where we will see different interpretations, and therefore this harmonization based on how 5.1 is written today, or actually more not how it's written, because it's clear how it's written, but how it will be interpreted.
Here's another non-clinical question. I'll ask that. In previous meetings, I've heard a lot about the cardiomyocyte studies and the in silico modeling, and those really seem not to have been given primary roles in the S7B revision. And do we have any insight into that? Well, that's a very good question. In the past 5 years, besides the use of concentration effect modeling, allowing us to do phase 1 QT assessments, there's also been a lot of talk about the so-called SIPA initiative and using cardiomyocyte assays, patch clamp data, and then integrating that in an in silico model to predict proarrhythmia risk. I don't know the details, but I suspect that there is not a uniform acceptance that these are really ready to go on a wide scale. We know that some research labs and institutions have done fantastic cardiomyocyte work, and there is some very fascinating modeling work, but I suspect that regulators just don't think that it can be widely applied across all sponsors and all labs at this time. So we may hear more about that in the next few years, but nothing, I think, is immediately on the horizon.
Can I add to this that Sure. It's clear also that Japanese researchers and also PMDA sees these myocyte assays with greater interest and probably with greater value than others do. So it's promoted in Japan and often used by Japanese companies.
And that leads to, here's a good question for you, Borje. The ICH previously set the threshold of concern that would lead to more phase 3 ECG monitoring as an upper confidence interval for QTC change from baseline greater than 10 milliseconds. But in the new Q&A, we see reference to a mean effect greater than 10 milliseconds being the threshold of concern. But there's then a very vague statement. The strength of the data depends on the upper bound of the two-sided 90% confidence interval. I wonder if you could comment on what that last bit might mean. You did address it a little bit in your presentation.
Well, I wish I could, but I'm not sure I can. I mean, it's obvious that a very narrow confidence interval gives you greater confidence in the estimated effect, but beyond that, I can't really decipher those two sentences. I know for sure that some regulators would prefer to retain or to keep the threshold being the upper bound below 10 milliseconds, and then make a case-by-case decision, because this is oncology, and it's clear if you have a life-saving drug with a huge unmet medical need, most regulators will still accept that the drug has a small QTC effect. So I don't think regulators are fully aligned on this, and I don't think that the renewed or the new criteria will help us in seeing harmonization across regulators. I can't give you more insight than so, Bob.
Here's another question about S7B that actually probably both of us can contribute to. At what point can a sponsor determine if the non-clinical studies really are adequate to support an integrated assessment of risk? Can you do that as you're completing the patch clamp work or the in vivo study? Or is that something that really has to wait later in the development process? So I'll start. That's a very important question because one would certainly love to know, as you're completing your patch clamp studies and your in vivo study, if you have really superlative data. The problem is you might have superlative data, but you don't have the clinical context in which to understand if it's complete. We know that to understand the risk of proarrhythmia of a new drug, you really have to know the clinical dose and exposure and intrinsic or extrinsic factors that will lead to the highest clinically relevant exposure. And until you know those, it's really very difficult to know if you have conducted your non-clinical studies to quote best practices. Borje, anything else to add on that?
No, not really, but I see a question that I think you should address, Bob. The question is, the threshold is 10 milliseconds in human therapy studies or in human studies. Are there any proposed thresholds for animal studies, such as non-human primates or dog studies?
That is a question that I have been asking myself as well, and that is not included in the S7B answers. And that's unfortunate because we're left hanging. And as I said earlier, there are people who do these animal ECG studies day in, day out and know them intimately. But then those same data are going to have to be reviewed by clinicians. And we're going to have to understand, was this a double negative study or not? And until we have better thresholds, I don't know how we're going to do that. Imagine if the E14 release did not say that the threshold of concern was an upper confidence bound of 10 milliseconds. Yeah, that's been debated and railed against for years, but at least we have a measuring stick to work with. If we didn't have that in E14, I don't know how anyone would have been able to know as they complete their clinical trials whether they had a QT concern or not. I think we're still at that point with the non-clinical QT studies. We need more guidance about what is the threshold of a positive versus a negative study, how high an exposure should be tested, what are the ECG guidelines, do you use implanted telemetry, do you use jacket, how do you measure the ECGs? All of these details that we've worked out for clinical ECG analysis really need to be laid out for us for the non-clinical studies as well.
Bob, I think we need to wrap up.
We're a little bit after the hour, so unless there are any other burning questions that anyone has, you have a few seconds to get them up. Otherwise, I want to thank everyone for joining us. This is a really important set of question and answers that I think is really going to be very helpful over the next few years.
And I can only agree, and I would also to reiterate what was said initially, that questions that have remained unanswered would be more than happy to come back with answers, or what we believe would be the answers. Thank you so much, also from me.
Thanks, everyone.